This conference will now be recorded.
Good morning, students. Thank you once again for coming for today's class. From today onwards, we're going to learn about virology. Okay, so today we're going to learn about general properties of viruses. First of all, what is virology? Virology is the bioscience for study of viral nature and the relationship between viruses and hosts. So, the study of virus is called as virology, and it usually deals with the nature of virus and how the virus usually has a relationship between hosts. History. Viruses are usually learned since a long back. Okay, do you know about smallpox? Yes, smallpox was endemic in China by 1000 BC. In response, the practice of viriolation was developed, or as vaccination. Recognizing that survivors of smallpox outbreaks were protected from subsequent infection, viriolation involved installation of dried crust from smallpox patients like snuff, or a later modification inoculation of pus from a lesion into the scratch or forearm of child. Okay, so smallpox was learned long back. Okay, so this smallpox usually involved to see the response of practicing viriolation. Viruses infections are universal. Viruses are present everywhere. It is present in our environment. They usually grow in our foodstuffs. They are present in animals, plants. Even there is a plant virology, animal virology, medical virology, which deals with humans and their association, virus and association. So, virology usually deals with them broad spectrum. Now, what is a virus? A virus is an obligate intracellular parasite containing genetic material which is surrounded by a protein. So, what is it? It is an intracellular parasite. Intracellular. So, for it to survive, it has to be present inside the living host. If the virus has to survive and multiply, it needs to be present in the tissues of the living host. Okay, these viruses are usually surrounded by protein material. Virus particles can be observed by an electron microscope. Recognizing the shape, size, and structure of tetrad viruses is critical to study the discipline. Yes, when you want to study a specific virus, you have to know its shape, size, as well as structure. Why structure? Structure is important because these are the parts where you can target the virus for virological purposes, for diagnostic purposes, for the preparation of vaccines. Okay. Viruses have an inner core of nucleic acid surrounded by a protein core known as apophyte. And most viruses range in sizes from 20 to 250 nanometers. Viral properties. Viruses are inert, filtrable agents. They are obligate intracellular parasites. This point is very important. What type of parasites are viruses? Obligate intracellular parasites. They cannot take energy or protein independent of the host. And they contain either RNA or DNA, but never both. Keep that in brain. Virus genetic material will be either RNA or DNA, but never both will be present in the same virus. Viruses have a naked capsid or envelope with attached proteins. Viruses do not have the genetic capability to multiply by division, and viruses are non-living entities. So size of virus. Can you see viruses? Viral structure, some terminology, some important definitions. Viral particle is usually called as virion. Keep that in brain. One viral particle is called as virion, or infectious, fully developed viral particle is called as virion. Now, what is the capsid? It is the protein which codes the genome, the genetic material. A protein which usually codes the genetic material is called as the capsid, and capsid are usually symmetrical. Okay, the capsid as well as the virion, that is the genetic material, is called as nucleocapsid. And nucleocapsid have an envelope or non-envelope. Viruses which have envelope are called as enveloped viruses. Which do not have envelope are called as non-enveloped viruses. Virion. So what is virion? It is a complete infectious unit of a virus particle. It is a with particle, and it is an extracellular virus particle. See, this is virion. A virus which has got a complete structure and it has become infectious. It is called as a virion. And parts of the virion, see, this is the complete structure. There is an envelope, there is the capsid, and inside the capsid there is a genetic material. Right? So everything is present. It is fully developed. So a fully developed infectious particle is called as a virion. How viruses are named? Viruses are usually named in different modes, in different forms. How? First one, based on the disease they cause. Okay, if they cause polio, they are called as poliovirus. If they cause rabies, they are called as rabies virus. Any other examples? If they cause measles, they are called as measles virus. If they are cause mumps, it is called as mumps virus. Okay, so depending upon the disease they cause, they may be. So that is the first one. Next, depending upon the type of the disease they cause, what type of disease are they causing? Are they causing cancerous disease? Are they causing gastrointestinal disease? Are they causing respiratory type of disease? So, depending upon this type of also viruses are classified. Example, murine leukemia virus. Murine leukemia virus. So what type of a disease this is? Leukemia means cancer. Next, depending upon the geographic locations, where it is distributed in the world. Example, Sendai viruses, Coxsackie viruses. Any other virus have you learned? There is one coronavirus. There is one coronavirus, one Middle East coronavirus respiratory syndrome, MERS. Understood? Middle East respiratory syndrome. While that coronavirus is called as MERS, because it's originated from there. Next, any other example? COVID. There has been often a conflict. Okay, there has been a conflict to name this COVID as Wuhan virus. Why they wanted to name it as Wuhan virus? Because it originated from there. So, depending upon from where the viruses had originated also, they can be. Next one, discoverers. Who has discovered it? Person discovering it. Depending upon that also, viruses can be named. Example, Epstein-Barr virus. Next, how they originally thought to be contacted, how they get contacted, whether they are contacted from egg or some spirit or by eating contaminated food. All of this. Example, dengue virus. What do you mean by dengue? Yes. Influenza, variant, influenza, we are all associated with influenza. Right? It is a common type of respiratory infection, influenza. Variant, why do we say it as variant? Because that usually spreads by contaminated things. Combination of all of the above. Example, drought, sarcoma, Next one, 5 basic types of viral structure. Can you see this? These are 5 basic types of viral structure. What are these? Icosahedral, enveloped icosahedral, complex, helical, and enveloped helical. See, icosahedral and enveloped icosahedral. Icosahedral, how many sites are there altogether? 1, 2, 3, 4, 5, 6. Envelope means icosahedral, which is surrounded by an envelope. Next helical. Next envelope helical and complex. These are the different viruses and their structures. Icosahedral. Different types of icosahedral viruses are adeno-associated virus, adenovirus, Coxsackie virus, cytomegalovirus, and hepatitis A, B, C, D, E virus. Herpes simplex virus, human immunodeficiency virus. Okay, these are some of the icosahedral viruses. Next. 3 types of capsid symmetry are there: cubic, helical, and complex. Baltimore classification system. Baltimore classification system is now the most important method of how you classify viruses for research purposes mainly. Depending upon these, viruses are classified into 7 types: double-stranded DNA virus, single-stranded DNA virus, double-stranded RNA virus, positive sense, single-stranded RNA, negative sense, single-stranded RNA, RNA reverse transcriptase virus, and DNA reverse transcriptase virus. Okay, so keep in brain altogether there are 7. So Baltimore classification is the present classification of viruses. 7 are there. Okay, first one is double-stranded DNA. Second, single-stranded DNA. Next, double-stranded RNA. Single-stranded RNA, 2 types are there: positive sense and negative sense. Next, reverse transcriptase viruses, 2 types are there: RNA reverse transcriptase virus and DNA reverse transcriptase virus. Okay, so this is icosahedral, or cubic symmetry. Icosahedral. Adenovirus, common example of icosahedral symmetry. This is helical. Have you seen this helical? Where it is? Tobacco mosaic virus. Okay. Helical. Helical is usually present in all of these viruses. Complex virus, pox virus. And what is an envelope? From where the virus obtains an envelope. Envelope is obtained by budding through a cellular membrane, except pox viruses. Possibility of exciting cell without killing them. And it contains at least one virally coded protein. Loss of envelope usually results in loss of effector. Classification of genome, that is either DNA or RNA. Okay, so these are all classification of DNA viruses. So DNA viruses, double-stranded, single-stranded, and complex. Double-stranded, enveloped or non-enveloped. If it is enveloped means herpes viridae. Non-enveloped. If it is non-enveloped, how is the structure? Whether it is circular or linear. If it is circular, then it is of two types. They are papilloma viridae and polyoma viridae. If it is linear, then it is retiniviridae. Single-stranded, non-enveloped, parvoviridae. Complex enveloped, box viridae. RNA viruses. RNA viruses are again divided into three types: single-stranded positive sense, single-stranded negative sense, and double-stranded. Single-stranded positive sense is of two types: enveloped and non-enveloped. Enveloped, what is the structure? Whether it is icosahedral or heptadral. If it is icosahedral, then they may be flavi, toga, or retroviridae. If it is helical, then they may be corona viridae. Next, single-stranded positive sense RNA viruses, which is non-enveloped, may be Pegorna viridae, Calciviridae, and Astroviridae. Next, single-stranded negative sense are always enveloped and helical. Example, Orthomyxoviridae, Paramyxoviridae, Rhabdoviridae, Filoviridae, Vania, and Arena. Double-stranded RNA viruses are Filoviridae. Okay. So keep all these in brain. Okay. DNA viruses. What are the DNA viruses? Keep in brain. P3, H2A, common code. Okay, what's it? P3, H2A. P3 means Foxviridae, Papovaviridae, Parvoviridae. These are three. Okay, Foxviridae, Papovaviridae, Parvoviridae. P3. Next, H2. Herpesviridae, Hepadnoviridae. H2. And 1A, that is Edenoviridae. Okay, DNA viruses. For P3, H2A. What are they? Foxviridae, Papovaviridae, and Parvoviridae. H2, Herpesviridae, Hepadnaviridae. A1, adenovirus. Next one, RNA virus. There are so many types of RNA virus. So you keep in mind. First of all, you come to me about DNA viruses. Okay, what's the code for DNA viruses? P3, H2A. Okay, once you know this, rest of all the viruses which are there, you keep them in RNA virus. Like that, you can easily know. So RNA virus, coronaviruses, orthomyxoviruses, paramyxoviruses, Chaga viruses, Flavi, Vania, Arena, Rhabdo, Rio, Corona, Retro, Calci, and Filoviridae. Okay. Next, basic steps in the life cycle. What are the basic steps in the life cycle of virus particles? Absorption, filtration, uncoating, and eclipse. Synthesis of viral nucleic acid protein. Assembly and release. So these are the steps in the life cycle of viruses. Okay. So first one is viral replication. It consists of virus attachment and entry. Next, uncoating of virion. Next, migration of genetic acid releases, transcription, replication. After transcription and replication, there will be translation. After translation, all the virus will come together. That is called as assembly. When all the virus particles are assembled, at last they are fixed. So, first one is, what is this? First one is attachment. Okay. For any virus particles cause infection, it has to first attach to the cell surfaces. That is called as adsorption. Adsorption is usually temperature dependent and requires viral attachment proteins and cellular response. Okay. Next, what will happen once the virus has attached? It will penetrate. Can you see the picture? Okay. So first one is attachment, then penetration, and once it has penetrated down, what is happening? The genetic material is entering into the cell. Penetration of enveloped viruses may be happen by two methods. That is fusion with plasma membrane or entry through endosomes. Fusion with acidic endosome membrane. See this one? Okay, so what is happening here? It is entering. After entering, the genetic material is being released. Next, virus take up through endosomes is called as viropexis or endocytosis or endocytosis. Penetration of non-enveloped viruses. Penetration of non-enveloped viruses usually takes place directly across the plasma membrane. See here, this is the non-enveloped viruses. It is not having any envelope. So how does it enter the cell? Directly by crossing the plasma membrane. Whereas in the case of these enveloped viruses, it has to first fuse the plasma membrane and then entering through endosomes. Whereas in the case of non-enveloped, no need of entry through endosomes. Replicative cycle. Since viruses are obligate intracellular parasite, virus must enter and replicate the living cells in order to reproduce. Yes, what are viruses? Obligate intracellular parasite. Therefore, it has to enter and replicate in the living cells. If the cells are dead, they cannot replicate. It has to reproduce, right? What are viruses? They are non-living entities. They usually need a living cell in order to reproduce themselves. This virus involves specific attachment of viruses, penetration and uncoating, nucleic acid transcription, protein synthesis, maturation, and assembly of virions, and the subsequent release from the cell by budding or plasma. Okay, now see this one. Okay, so what did you understand here? See first, the viruses get attached. Then, after attaching, it is penetrating. Next, after penetrating, what is being happened? Uncoating and release of the genetic material. And this genetic material usually undergoes replication, transcription, and translation. Okay, newly formed proteins such as regulatory proteins, structural proteins, all total proteins will be formed. Next, all of them assemble together. After all of these assembled together, what will happen? They will run off. Okay, now what is unfolding? Need to make the genome that is but once unfolding takes place, they usually enters into active phase. Active phase usually lasts until the first virus particle is formed. So what is unfolding? After the viruses have entered into the body, after they penetrate into the cell, unfolding takes place and the genome usually enters into the cell. Okay, this period when the genome enters into a living cell and replication takes place until a new virus particle is formed, that period is called as active phase. Synthesis of viral nucleic acid. Nucleic acid may be made in the nucleus or in the cytoplasm, whereas protein synthesis is always in the cytoplasm only. Assembly and maturation. Assembly and maturation that usually takes place at 3 sites. That is, in the nucleus, in the cytoplasm, or at the cell membrane. Phyllis. Now what is phyllis? After all the progeny virus particles are formed, they usually assemble together. There is lysosomes, and these newly formed virus particles usually bud through plasma. Not every released virion is infectious. Okay, so this is how viral replication takes place. Okay, so viral replication consists of attachment of virus. After attaching, they have to penetrate. After penetrating, there is unfolding and the genetic material enters into the cell. Next, there is transcription, translation, protein synthesis, and maturation. After this, what will happen? All the structural proteins and other proteins which will assemble together to form virions. And after they become mature, they usually rise and come out of the cell by budding or lysis. Next, transmission of viruses. How viruses infections are transmitted. Example, first one, respiratory transmission. Example, influenza virus. Have you heard about influenza? Yes, we usually call it as flu. Next, fecal oral. Example, enterovirus. Bloodborne transmission. Hepatitis B virus. Sexual transmission. HIV virus. Animal or insect vector. Rabies virus. Viruses enter the body of host in a variety of ways. Yes, viruses enter in the body of host in different ways. So what are those different ways? The common forms of transmission are first, inhaled droplets, sneezing, coughing. For example, common cold or influenza virus. The most commonest form of transmission of viral infection is through inhalation. It may be by droplets or viruses. Example, common cold, influenza. We all are getting common cold, right? In a year, at least 3-4 times we get common cold, right? Next one. By drinking water or eating raw food, for example, hepatitis virus, poliovirus, poliovirus. What type of virus is poliovirus? Fecal oral transmission, poliovirus belongs to enteroviruses. Next one. Other modes of transmission by sexual intercourse, for example, for HIV and hepatitis B. Also, vertical transmission from mother to baby. Yeah, from mother to baby. That is also possible. HIV, hepatitis B, and rubella. Also, bulbs of vector arthropods. Dengue fever. Have you heard about this? Right. Very good visuals. Right? Others, yellow fever, Rift Valley fever. Okay. So, paralysis infection can also be transmitted by bites of arthropod vectors. However, most viral infections do not lead to serious complications, and the host gets well after a period of sickness to be removed for the rest of their lives. For example, measles infection, rubella, or general measles, mumps, and okay. So these are the different modes of viral infection: inhalation, oral fever, transmission, sexual, or disease. Okay. Some viral infection, once we get, they will usually give immunity to our body from example, measles infection, rubella. Next, cultivation of viruses. Cultivation of viruses is very important. Please learn this. Cultivation of viruses. First of all, why do you need to cultivate viruses? Why are you cultivating bacteria? In order to isolate the organism. Right? Similarly here also, when you want to learn which is the virus which is causing a particular disease, you need to cultivate them. Also, cultivation of virus is not only important to identify the virus, but also important in modern creation of vaccines, etc. There are three main systems of cultivation of viruses. First one is animal inoculation, next embryonated eggs, and tissue cultures. Tissue culture is of three types: organ culture, explant culture, and cell culture. Animal inoculation. Different types of animals are used for experimental purposes, such as mice, fat-sucking mice, rats, chickens, monkeys, and pigs. Growth of the viruses in inoculated animals may be indicated by death, disease, or virus. So, you are inoculating into animals. How can you identify the growth of the virus? By seeing if there are any lesions on the experimental animals, or if there is death or disease in the animal which you have used. This animal inoculation is mainly used for the study of pathogenesis, immune response, epidemiology, and pathogenesis. Disadvantages of animal inoculation are that immunity may interfere with viral growth and that animals often harbor latent viruses. Next, embryonated eggs. What are these embryonated eggs? So embryonated eggs. The embryonated egg offers several sites for the cultivation of viruses. So this is embryonated egg. Different sites are there where we can inoculate the viruses, such as chorioallantoic membrane, amniotic cavity, box sac, allantoic cavity. Okay, any all the sites you can inoculate. Let's see for the presence of Inoculation interchorioallantoic membrane produces visible lesions. Pox. This is mainly used for the cultivation of variola and vaccinia virus. Inoculation of allantoic cavity is used for influenza viruses and paramyxoviruses. This type of method is also used in vaccine production. Inoculation into amniotic cavity is used for the primary isolation of influenza virus. Drop sac inoculation is used for the cultivation of some viruses, chlamydia as well as syphilis. This diagram very important. All of you have to learn this diagram. Please learn to draw it. Pencil. So smallpox in chorioallantoic membrane of embryonic egg. This is chorioallantoic membrane. Can you identify the pox? They are fluorescent, like dots, white dots. Next, tissue cultures. Tissue cultures are of different types: organ culture, explant culture, and cell culture. What do you mean by organ culture? Small bits of organs are usually maintained for several days to weeks. Okay, this type of culture is mainly used for the isolation of viruses which appear to be highly specialized, such as tracheal ringworm culture, coronavirus. Next one, explant culture. Fragments of loose tissue can be grown on explants emitted in plasma clots. Example, adenoid tissue explant viruses for adenovirus. Okay, so first one is organ culture. What is this organ culture? You take small bits of organs and you maintain inventory for several days or weeks. Okay, the original architecture and function of the organ is usually maintained. Most common type of organ culture is used for coronavirus we are using, that is tracheal ring culture. Next one is explant culture. In the case of explant culture, fragment of loose tissues, the tissues are usually grinded and then blown on explants together with plasma. Cell cultures. All the embryonated eggs and laboratory animals are very useful for isolation of viruses. Cell culture is the source system for virus isolation in most laboratories. This among the embryonated egg and missing of laboratory animals. Cell culture is the most commonly used method for laboratory diagnosis. We use special cell lines. These cell lines consist of viral media. In viral culture media they contain, and we usually maintain those on these based on their origin. Chromosomal characters and the number of generations which they can be maintained. Cell cultures are divided into three types. They are primary, diploid, and continuous. So what do you mean by primary? Primary means they are directly prepared from animals or fish. Okay, a culture, a cell line, which is directly prepared from an animal or human tissue, is known as primary cell cultures. And such type of cell cultures can be subcultured only once or twice. Example, monkey kidney cell, human embryonic kidney cell. Mainly used for isolation of viruses and for vaccine protection. Next one, diploid cell cultures. Diploid cell cultures are derived from human fetal tissues and can be subcultured 50 to 50 times. Mainly useful for the isolation of ascaris pathogens. Example, polio virus, rubella virus, rabies virus, etc. Continuous cells. These are cells of single time. Continuous cell lines are usually derived from past cells and are capable of continuous serial progression. There are so many types of continuous cells, such as HELA, HeLa, H2, Vero, McCoy cells. Next one, detection of virus growth in cells. How will you detect the virus growth in cell culture? Okay, most commonly we are using cell cultures, right? How will you detect this? Many viruses cause morphological changes in cultured cells, and these changes can be readily observed by microscopic examination, and these changes are known as cytokines. Okay, now what happens is we are using cell lines, right? When a virus grows in the cells, the cell structure changes. That is called as cytopathic effect. And cytopathic effect may be cytosolic or non-cytosolic. Means when there is a change, the cell may die or may not die. Okay. Non-cytosolic effects include eosinophilic or basophilic inclusion bodies of the nucleus or cytoplasm. Cytopathic effects can be so characteristic of individual viruses that they are mainly used for identification of virus. See this one. Normal cells. Can you see? Cytopathic cells. First, down one A. Cytoplasmic inclusion bodies caused by rabies virus. B. Syncytium formation formed by the cell fusion due to infection with measles virus. C. For measles virus, syncytial formation. Why you are calling it syncytial? All the cells are coming together and forming a group, a large group that is called syncytial formation. So there are different forms of cytopathic effect. Okay, the cells may become round, the cells may become crumbled, or they must more than their normal size, or all the cells can be glued together to form a syncytium. These are the different types of cells. Last one, viroids and prions. What are viroids? Which is a single-stranded RNA genome and smallest pathogen, mainly expressed. Prions, infectious particles that are entirely clean and non-nucleotide, highly heat resistant. Okay, and these prions usually cause a number of infections and mainly affect the nervous tissue. Example, bovine, spongiform encephalitis, also called as mad cow disease, scrapie in sheep, kuru, and Creutzfeldt-Jakob disease in humans. Okay, so that's all for today. Thank you all of you for attention. From today's chapter, please learn different types of viruses, classification of virus, what are DNA virus and what are RNA virus. Next, cultivation of viruses. What are the three modes of cultivation? Animal inoculation, impregnated egg, and cell cultures. Different types of cell cultures. And next one is how will you demonstrate growth in cell lines to see the presence of cytoplasmic spread or. Okay, thank you once again for today's class.